S., a woman in her 60s, recently suffered from a stroke to the right cerebral hemisphere, and while her intelligence remains intact, something is gravely wrong. She cannot see anything placed to her left. The idea of left has evaporated. When she puts on lipstick, she covers only the right half of both lips. Rather than turn to the left, she turns to the right and goes all the way around until she has reached her intended position. If a dessert is placed on the left side of her tray, it goes unnoticed until a nurse gently turns her head to demonstrate its existence. Her stroke has gravely affected the way she perceives the world, her senses. You can think of your sensory organs as the brain's window to the outside world. Through the process of transduction, your sense organs turn external stimuli, like a flashing light, into electrical signals to be processed by the brain. Did you know that your eye works like an old-fashioned camera? Let's dive into the complicated processes involved with vision. Light passes through the cornea and enters the eye through the pupil. The iris regulates how much light enters, and the lens bends the light so that it focuses on the retina, the inner surface of the eyeball. What you see is mapped onto the retina as a reversed image. And then the signals travel through the optic nerves to other parts of your brain. The retina has 3 types of neurons: photoreceptors, interneurons, and ganglion cells, organized into layers. Ganglion cells and interneurons relay information from the photoreceptors, rods and cones, which are found in the periphery of the retina. Rods allow you to see in dim light, while cones pick up detail and color. The center of the retina, the fovea, are where cones are the most densely packed, which is why the vision is sharpest there. Around the fovea is an area called the macula. Signals from photoreceptors are processed and sent to the brain via the optic nerve. Signals converge at the optic chiasm, where fibers from the left side of the retinas continue to the left brain and fibers from the right side go to the right brain. This crossover ensures that visual information from each eye reaches both hemispheres of the brain. So when S had a stroke, any visual pathways on the right side of her brain were demolished. Thus, fibers from the right side of each retina were rendered useless, and so she could not view anything to her left. Visual signals pass through the lateral geniculate nucleus of the thalamus, then to the primary visual cortex in the occipital lobe, where the retina's visual map is preserved and further processed. The primary visual cortex, a thin sheet of neural tissue, is located in the occipital lobe at the back of your brain. It has several layers with densely packed cells. The middle layer receives direct input from the thalamus, retaining the retina's visual map. Layers above and below interpret more complex features like edges, angles, and motion. These layers allow the brain to identify specific orientations, shapes, and movements, enhancing her ability to perceive detailed and dynamic scenes. Visual signals are fed into several parallel but interacting processing streams. The ventral stream, or the what, which heads towards the temporal lobe, processes information about an object's shape and color, integrating it with memories to identify objects. For example, recognizing your neighbor's dog based on its appearance. The dorsal stream, or the where, moving towards the parietal lobe, deals with spatial relationships, motion, and timing, helping you navigate and interact with your surroundings. This allows for unconscious actions like shouting stop without thinking. Now, you may be wondering, how can we see in 3D if visual input is mapped onto 2D? Binocular vision enables depth perception by merging slightly different images from each eye, creating a 3-dimensional view. This is essential for perceiving depth accurately. Because nerve fibers cross over at the optic chiasm, each side of the cerebrum processes information from the opposite visual field, allowing you to see the full picture. Research has shed light on many different visual disorders. Insights from animal research have improved treatments for strabismus, or crossed eyes, allowing for early correction and preventing permanent vision loss. Also, macular degeneration, a major cause of blindness, is being addressed through gene and stem cell therapies. Researchers are also exploring ways to send electrical signals directly to the brain via ganglion cells to bypass damaged photoreceptors. From the initial capture of light to the complex processing in the visual cortex, each step in this journey reveals the amazing mechanisms that enable us to perceive the world around us.
Of course, when you think of sight, your next thought is sound. You're listening to this video right now. You're also probably listening to cars outside, or maybe a thunderstorm, or one of your parents talking to someone else. Maybe someone's even talking to you, and you haven't noticed yet because you're so riveted by this video. That's all your brain. Hearing allows you to communicate with others by processing and interpreting complex messages in the form of speech sounds. Your brain records a sound's pitch, loudness, duration, and location, and it does this for everything you hear. It even breaks complex sounds into separate components or frequencies. As a result, you can follow particular voices in a conversation or instruments as you listen to music. You'll notice this if someone talks to you while you're watching the video. Your brain separates out the sounds so you know that there are two different speakers. In order to do all of that, your ears are precisely built for their function. Sound in the form of air pressure waves reach the outer ear, where waves are funneled into the ear canal to reach the eardrum, also known as the tympanic membrane. The eardrum vibrates in response to these waves, sending vibrations to three tiny bones in the middle ear, the malleus, incus, and stapes. The stapes pushes on the oval window, a membrane that separates the air-filled middle ear from the fluid-filled cochlea of the inner ear. The oval window converts the mechanical vibrations of the stapes back into pressure waves in the fluid of the cochlea, where specialized receptor hair cells convert the pressure waves into electrical signals. So, how does all this physical information get to the brain? Hear me out. This is where the basilar membrane comes in as it vibrates in response to the pressure waves. It lines the cochlea in a spiral pattern, and much like an instrument, it's tuned across the spiral ramp. Think of it as a trampoline, where the outer edges are tighter and vibrate in response to high-pitched sounds, while the middle is much bouncier as it vibrates to lower-pitched sounds. On top of the basilar membrane sit hair cells. These cells are extremely important in hearing, and damage to these hair cells is the main cause of hearing loss. These hair cells are tipped by stereocilia that brush against the tectorial membrane. These tiny stereocilia are kind of like the strands on a broom and all brush against the membrane. This motion opens up small channels that lets ions in and transduct this physical information into an electric signal. Each hair cell only responds to a certain range of frequencies, adding to how our ear can be tuned to different sounds. These ions help excite the auditory nerve, which carries information to the thalamus, the brain's relay center, or mailman. This mailman can then send the information to the auditory cortex in the temporal lobe for more processing. The temporal lobe is extremely important in processing auditory information. I like to remember this by the way that we often put our fingers to our temples when we're trying to focus on a certain sound, or when singers will touch their temple to hear themselves better. A really cool thing about the auditory cortex is that it preserves all the information from the basilar membrane about the pitch and frequency of the sounds. So when it reaches the auditory cortex, we are still able to separate the sounds and even have specific neurons to differentiate the intensity, duration, harmony, and rhythm of those sounds. It's how you know that it's your mom talking to you, or that it's your friend yelling at you from across the cafeteria. Next up is taste and smell. Believe it or not, these two senses actually go hand in hand. Aside from the fact that they both regularly regenerate, with olfactory neurons being the only sensory neurons that regenerate through our life, what we consider as taste is actually mostly just smell. 80%, actually. Taste comes in 5 forms. There's the classic ones like salty, sour, sweet, and bitter. The one that most people don't recognize is umami, which is Japanese for savory. Contrary to popular belief, they are not divided into 5 sections on our tongue. They are actually evenly spread out. Taste has an interesting path to the brain, as it starts when molecules bind to receptors in the tongue. Then, similar to the other transduction processes, physical turning into electrical, 3 cranial nerves take this information to the mailman, or the thalamus. The thalamus routes it to the gustatory cortex in the frontal lobe and insula, which sits under the cortex.
Smell works similarly, as odor molecules will bind to receptors and excite the olfactory nerves, which carries information directly to the two olfactory bulbs and primary olfactory cortex on the underside of the temporal lobe. Did you see a star player missing in that lineup? That's right. Olfaction is the only sense that goes directly to the corresponding cerebral cortex without passing through the thalamus first. Similar to hearing, the information about the type of smell is preserved and transmitted to the appropriate cortexes. It's why we can taste and smell so many different things, despite only having around 1,000 types of olfactory cells. So why do I keep putting taste in quotes? Well, that's because what we consider taste isn't the real version of taste. Taste and smell are so linked that they actually work together, always. For example, have you ever had a cold that stuffed up your nose so everything always tasted bland? Or have you noticed that sugar tastes sweeter when mixed with the smell of strawberries? In fact, there are some cells in the frontal lobe that respond to just smell and taste combos. Talk about specificity. Finally, we have the somatosensory system, which is responsible for touch sensations. This can be a variety of things, all the way from light touch to texture to pain. We perceive these sensations with touch receptors located in different layers of the body's main sense organ for touch. Touch receptor signals travel along sensory nerve fibers to neurons in the spinal cord. From there, they go to the thalamus and somatosensory cortex, where the signals are processed into perception. There are two types of nerve fibers for touch information, especially pain and itch messages: fast A-beta fibers, which have thick myelinated axons, and slow C fibers, which have thin unmyelinated axons. Different parts of your body vary in sensitivity. For example, the lips and fingertips have more receptors per unit area than your back. Measuring sensitivity in receptors per unit area is called two-point discrimination, the minimum distance between 2 points on the skin that a person can identify as two distinct stimuli rather than a single stimuli. Pain, a warning signal that consists of both a sensory and emotional aspect, occurs when special sensory fibers called nociceptors respond to stimuli that can cause tissue damage, acting as a defense mechanism. There are different types of nociceptors for different types of stimuli, from itchiness to chemical stimuli. Surprisingly, nociceptors also respond to chemicals in spicy food, like capsaicin in hot peppers, producing a burning pain. Tissue injury releases many chemicals at the site of damage to cause inflammation, which allows you to continue feeling pain, acting as yet another defense mechanism. Neuropathic pain is a state of hypersensitivity to pain caused by a malfunctioning nervous system. Lipids, called prostaglandins, increase your sensitivity to tissue damage, allowing you to feel pain. Interestingly, prostaglandins also contribute to a type of neuropathic pain called allodynia, a condition resulting in a hypersensitivity to pain. For example, even a soft touch can produce a pain. Another example of neuropathic pain is diabetic neuropathy, where nerves in the hands or feet are damaged by long-term exposure to high blood pressure, resulting in a variety of symptoms from numbness to aching pain. As mentioned earlier, fast A-delta fibers evoke immediate sharp pain like from a pinprick, while slow C fibers cause a dull and diffused ache over a larger area. But then, why do different people experience the same pain differently? Well, this depends on a person's emotional state and setting in which the pain occurs. Pain messages are processed in a region of the brainstem called the periaqueductal gray matter, or PAG, which modulates pain and activates certain pathways that release endorphins. Endorphins, opioids produced naturally by the body, can cause feelings of happiness. Endorphins act similarly to the analgesic morphine. Analgesics are drugs that relieve pain without a loss of consciousness. Similarly, adrenaline produced in emotionally stressful situations can also work. Endorphins, opioids produced naturally by the body that can cause feelings of happiness. Endorphins act similarly to the analgesic morphine. Analgesics are drugs that relieve pain without a loss of consciousness. Similarly, adrenaline produced in emotionally stressful situations can also work as an analgesic. Endorphins and adrenaline help the body regulate and reduce pain. Scientists are constantly researching and finding new methods to control pain, especially for individuals afflicted with chronic pain. Some examples are injecting analgesics and targeting the painful stimulus itself. Other methods involve targeting the emotional aspect with meditation, hypnosis, massages, and cognitive behavioral therapy. And surprisingly, research has revealed that cannabis can suppress activity in a few pain areas in the brain, reducing the emotional toll that chronic pain takes.